Hello everyone, this is Fanindra, Zoology faculty. Now let us discuss a topic called biotechnology and its applications. In this, when we go with the zoology part, we have the biotechnological applications in the medicine. So in the biotechnological applications, like the recombinant DNA technology have produced enormous amounts of products and which are very safe and which are more effective therapeutic drugs. Therapeutic drugs, the drugs which are used in treatment, they are called as therapeutic drugs. So in this effective therapeutic drugs, a very important point which you have to remember, these are isolated from non-human, they induce, because the recombinant therapeutics do not induce any unwanted immunological responses, because the drugs which are been, the chemicals which are been extracted from the non-human sources or any other thing, they can cause some unwanted immunological responses, but the recombinant therapeutic drugs will never cause any unwanted immunological responses or some sort of allergies or any other reactions. At present, about 30 recombinant therapeutics have been approved for human use, and in the world, around India, 12 of these are presently being marketed. So these are the points which are given in NCERT, so you have to remember as it is. 30 recombinant therapeutics around the world, but in India, 12 are presently being marketed. Okay. Now, in this, when we go with the first thing, the genetically engineered insulin. Now, what does an insulin do? Insulin helps mainly in treatment of diabetes mellitus, because insulin generally, what does insulin do? Insulin will convert glucose to glycogen. So, it reduces the glucose level in our body. So, but when there is a deficiency of insulin in our body, the glucose level gradually increases, and that results in a condition called as a diabetes mellitus with excess glucose present in our body. So, for that, we are like with the people who are suffering from the diabetes mellitus, we are generally taking the insulin injections, so that because these insulin injections, when the insulin level increases in the body, then automatically the glucose level gradually decreases. So, where do we get this insulin? Now, why don't we take insulin through our mouth? Because it's a protein which gets digested in our HCl, so insulin can never be taken through the mouth. So, there are some oral administered insulins which are being developed, but now as per NCERT, we are not discussing about those things. As per NCERT, we are taking insulin injections to reduce or to treat diabetes mellitus. Insulin is used in diabetes. Earlier, it was extracted from pancreas of slaughtered cattle and pigs. From killed cattle and pigs, the insulin was being extracted. But as it is from an animal source, this insulin causes unwanted immunological responses, obviously. Now, this insulin, if you talk about, if you look at the structure of insulin, insulin is made up of 3 chains here: A chain, B chain and the C chain. A chain and B chain and C chain totally, A chain is made up of 21 amino acids and B chain are made up of 30 amino acids, altogether 51, and then C chain is made up of the remaining amino acids, totally forming 33 amino acids, totally forming 84 the different types of amino acids which are present here. The A chain and B chain, the insulin, if you look at it, this is proinsulin and this is insulin. Proinsulin is something which is the pre-insulin or proinsulin, which has to be modified to insulin, so that this insulin is acting on our sugar to convert into glucose to convert into glycogen. So, how to convert this proinsulin into insulin? Proinsulin, if we remove the C peptide from this proinsulin, the C peptide, if it has been removed, then automatically the proinsulin will be converted into the normal insulin, right, which is inactive. So, to remove this C peptide from this insulin is been removed, so this proinsulin converts into insulin, which is called as a maturation of the insulin. If you look at it, in mammals, insulin is synthesized as a prohormone. What is prohormone? Like a proenzyme, it is an inactivated hormone, and then this prohormone needs to be processed. How it is been processed? It is processed by removal of C peptide, and this C peptide, if it has been removed, then it becomes the normal mature insulin. Now, when the people are producing insulin using this recombinant DNA technology, using the E. coli, the main challenge for this is to produce generally they can produce A chain and B chain, but the main challenge between here to produce a mature insulin is to join the A chains and the B chains. It is a very important risk and challenge to these people, because A chain and B chain are joined together with the help of two disulfide bridges, and there is one disulfide bridge present on the A chain. It's an intra-disulfide bridge, and these two are inter-disulfide bridges. So, in 1983, Eli Lilly, an American company, prepared two DNA sequences corresponding to A and B of human insulin and introduced them into the plasmids of E. coli to produce two insulin chains separately. When these two chains A and B were produced separately, they were extracted and combined, by using this disulfide bridges, and the human insulin was being prepared artificially by using this recombinant DNA technology by Eli Lilly, an American based company. Okay, this is about the genetically engineered insulin, or it is also called humulin, which never induce any unwanted immunological responses. Okay. Then the next application in biotechnology is gene therapy. What is therapy? Nothing but the treatment. See, for example, a person is born with a hereditary disease. Can a corrective therapy be taken for such a disease? For example, a person is suffering from a genetic disorder. Is there any therapy to treat this genetic disorder? Okay. Gene therapy is an attempt to do this. There are some diseases which can be treated with the help of this gene therapy. So, what is gene therapy is doing? It is helping in correction of a gene defect which is diagnosed in the child or an embryo. Here, the genes are inserted into the person's cells or tissues to treat this particular disease. Or sometimes the gene correction of a defect of a normal gene into an individual or to replace this non-functional gene with a normal gene also helps in the correction of the gene, which is nothing but the gene therapy. For this, if you take an example for gene therapy, the first clinical gene therapy was done in 1980 to a 4-year-old girl she was suffering from the deficiency called adenosine deaminase deficiency, ADA deficiency. What is the use of this enzyme? This is very crucial for immune system to function. Because so if immune system fails to function here, then again this girl is going to suffer from many diseases which is called SCID, severe combined immunodeficiency. Severe combined immunodeficiency because of the non-functional enzyme which is called as ADA, adenosine deaminase enzyme adenosine deaminase deficiency. So this is very clearly given here, caused due to the deletion of a gene for adenosine deaminase. If there is a deletion in that, and it causes the non-functional enzyme or may not produce any enzyme. In some children, ADA deficiency can be cured by bone marrow transplantation. In others, it can be treated by enzyme replacement therapy. Sometimes bone marrow transplantation can be done. Sometimes enzyme can be given to the baby or given to the child. Enzyme replacement can be done. But these both are not permanent. They are only for temporary cure. But both the approaches are not completely curative. Now, what we are going to do? Here, there is one more method. Here, in the step towards the gene therapy, we are going to extract the lymphocytes of the baby, of the patient, and we are going to culture the lymphocytes outside. And after culturing the lymphocytes, a functional ADA cDNA, complementary DNA, which has been prepared by using a retroviral vector, is inserted into these lymphocytes, and these lymphocytes is again inserted back into the patient. Okay. Now, these lymphocytes will start producing this ADA enzyme, but the lymphocytes are not immortal. Very important, a previous NEET question, not immortal. What is meant by immortal? Immortal is something which there is no death at all, but lymphocytes will die. So, as these lymphocytes are not immortal, these cells keep on dying. Now, when all the lymphocytes die, again a periodic infusion of new lymphocytes should be done. Periodic infusion, what is infusion? Again, these genetically engineered lymphocytes are inserted back to the patient continuously, periodically. It's not like if once we insert these genetically engineered lymphocytes throughout the lifetime, it can protect the children. No, it's not possible. So, because the lymphocytes are not immortal, it can be an assertion reasoning question. Okay. So, that's the reason periodic infusion of lymphocytes has to be done to keep the baby healthy. But if this same gene isolate from the marrow cells producing ADA is done at early embryonic stages. Early embryonic stages, if this process is done, then it can be a permanent cure. It could be a permanent cure. So that's about the gene therapy ADA deficiency enzyme. Okay. The next comes the molecular diagnosis. So what is meant by diagnosis? Diagnosis is nothing but an identification and of a particular disease. First you treat a disease and then you should identify the disease. Okay. So for the treatment, early diagnosis is very, very important. But once upon a time, conventional methods are the traditional methods. These conventional methods are used to identify the disease by the serum analysis and the urine analysis. But in the serum analysis and urine analysis, early detection is not possible. Because why do we go for serum and urine analysis? When we get the symptoms outside. If we get the symptoms outside, it means that already a pathogen has divided continuously in our body. So we cannot, once the symptoms appear, it means that already the disease has spread rapidly or widely. So going to the blood test or serum urine test or at that particular time, we cannot reduce the disease unless and until we take medicines. So how to identify it in an early stage? There are some methods using recombinant DNA technology, PCR, polymerase chain reaction, and as well as enzyme linked immunosorbent assay. By these methods, early detection of the disease can be done. So when we go with the PCR, presence of pathogen, any bacteria or viruses, is normally suspected only when a pathogen has produced a disease symptom, as I told you. But once a symptom appears, in the sense already the disease causing pathogen already divided much rapidly in our body. But you can identify this pathogen in a low concentration with the help of PCR, because PCR helps in amplification of their nucleic acid. So when it helps in amplification of this nucleic acid, even a small amount or any pathogens present in our body, we can identify with the help of a specialized technique called as PCR. So PCR is used even in identifying the HIV in the AIDS patients. Like once the virus enters, we can easily identify. PCR is also used to identify the mutations in the cells of the cancer patients. PCR is also used to identify many genetic disorders. Next to this, we can go for the single standard, that is probe autoradiography. So what is autoradiography? In autoradiography, we are taking a DNA probe or DNA or RNA probe. For example, we have to identify a cancer, a normal cell. We'll take a normal cell, we'll take the DNA or RNA. In the normal cell, the DNA or RNA is made into fragments, and then radioactive materials are added to these DNA fragments. Now this is called as a probe. Now, we'll take a cancer cell, we'll take a cell which is suffering from the cancer. Now, again we'll take the DNA, we'll cut the DNA into fragments. Now, after adding them into agarose gel electrophoresis, we hybridize these DNA with this probe, because these probes, when we are adding to this particular DNA, this probe may come and join with this particular DNA because it has a complementary base pairs. But there are some locations where this probe may not join with this DNA, because this DNA, there are some mutations which are occurring in this DNA. If because of these mutations, the sequence of the DNA has been changed so that this probe could not join. So if the probe could not join, we can easily say that this part of particular DNA is having some mutations. Now this can be observed easily on the autoradiography and X-ray photographic film, because these are attached with the radioactive molecules. So we just take an autoradiography of a normal cell, we just take an autoradiography of a cancerous cell, we can find the difference in the bands between the normal cell and a cancerous cell. So that is how we can also detect the mutations in the cancer cells in the body with the help of this autoradiography technique, by using the RNA or DNA, single standard DNA or RNA as a probe which is coated with a radioactive molecule. When it comes to ELISA, ELISA is based on antigen-antibody interactions. So based on the antigen-antibody interactions, mainly antigens like proteins, glycoproteins, etc., and their interactions with the antibodies, this ELISA can be assessed. So mainly ELISA is used to test, it's a diagnostic test for HIV or AIDS. So these are all the techniques how we can just identify the pathogens at an earlier stages rather than the symptoms appear in our body. Because if we identify the pathogen at an early stage, there are some disorders like AIDS which cannot be cured, but we can increase or prolong the lifespan of those individuals if we detect the pathogens so early by using the different types of techniques like PCR and the autoradiography technique and as well as the ELISA techniques. These are called as molecular diagnosis. Okay. Next comes to transgenic animals. What are trans? Trans is nothing but change, you all know that. Transgenic animals, so genes are being changed here. So animals have their DNA manipulated and process an extra foreign DNA are known as transgenic animals. So their DNAs are being manipulated or a foreign gene is being inserted into the animal. Now that will become a transgenic animal. So there are many different types of transgenic animals. Rats are there, rabbits, pigs, sheep, cows, fish, all these are the different types of transgenic animals. Although over 95% of the transgenic animals are mice. 95%, all mostly used animals are mice. Now, why these animals are being produced? What are the uses of these transgenic animals? The first thing is normal physiology and development. Normal physiology and development. Now, how to know that this particular gene is assessed for this particular function? A gene is performing some particular function. See, for example, you will take a gene or you will take something which affects this particular gene in a cell. Now, the functioning of that particular organism will be disturbed. So, we can study the disturbance of this functioning in the organism mainly in the transgenic animals. If you look at it, transgenic animals are specifically designed to allow study of how genes are regulated and how they affect the normal functions of the body. For example, insulin-like growth factor. By introducing genes from other species which alter the formation of this factor, I am disturbing this insulin-like growth factor by introducing some genes. When insulin-like growth factor is being disturbed, now what functions in the mice are being disturbed? That can be analyzed. So, by that we can say that these are the functions which are related to insulin-like growth factor. So, that is how you can study the normal physiology and development. Okay. Then, study of the disease. Now, transgenic animals are mainly used to study how the disease occurs, like what happens when a disease occurs, what are the changes that are occurring in the body, what kind of medicines can be invented. All these can be done with the help of these transgenic animals because these are acting as a transgenic models. So, for many human diseases, mainly like cancer, cystic fibrosis, rheumatoid arthritis and Alzheimer's, there are models for the transgenic models for the identification and cure for the disease. Then biological products, we can obtain so many biological products from the transgenic animals. So, there are some products which are helping to treat the diseases. There are some products which increases the protein content. There are some products which are acting as chemicals, many things, nutrients, many things. So, mainly if we take the products, biological products, mainly by changing the gene of that particular animal, changing the gene and introducing the gene into a particular animal or any animal, this animal can produce the different types of products. For example, anti-human protein alpha-1 antitrypsin which has been produced, which is used to treat emphysema. What is emphysema? The emphysema is a respiratory disorder where the alveolar walls get damaged because of tobacco smoke. Right. Similarly, there are attempts made to treat phenylketonuria and cystic fibrosis. In 1997, the first transgenic cow, Rosie, has been produced. Now, this Rosie will produce a human protein-enriched milk 2.4 grams per liter. Generally, it is very less, but this Rosie will produce a milk with 2.4 grams per liter, which has human alpha lactalbumin, which is a more balanced product for the human babies than the natural cow milk. So, this is how we can produce many biological products with the help of these transgenic animals. Then vaccine safety. Transgenic animals are used to test for vaccines. So, because when you discover the vaccines, these vaccines are tested on the transgenic animals. Mainly transgenic mice are used to test the safety of polio vaccine. If this is reliable, if the testing on mice is successful, then we can replace with the use of monkeys, because testing with monkeys it's not environmental friendly. We are killing the monkeys and then it is a very costly thing. Economically, it is very high. So, if you use transgenic mice and if it is successful, we can replace the use of the monkeys and we can directly test on the transgenic mice. That's about vaccine safety. Chemical safety testing, chemicals, that is nothing but toxicity testing or safety testing. So people discover so many drugs, people discover so many medicines and so many chemicals. So these drugs, before trying on the humans, these drugs are tried on the transgenic animals, and if they are successful, they will try on the humans. Human trials can be done. So that's how the chemical safety testing can be done, because these transgenic animals, very, very important point, these transgenic animals carry the genes which make them more sensitive to toxic substances than the non-transgenic animals, because non-transgenic animals will not be so sensitive. It takes a lot of time to produce a result, whereas transgenic animals are more sensitive. As they are more sensitive, immediately when the drug is injected into transgenic animal, it can produce a result immediately. When the result is produced immediately, then it is very easy to test the drug. So that's the reason transgenic animals are mainly used to study the effects of these chemicals or toxins which are, or the drugs which have been discovered by the humans. So this is how we are using transgenic animals for normal physiology and development. We are using it for study of diseases. We are using them for producing so many biologically important products. We are using them for vaccine safety, vaccine testing, and we are also using them for chemical safety or toxicity testing. So this is how, these are the topics which we study in biotechnology and applications in medicine from the zoology part. The remaining part you will study from the botany part. Okay, then. So now the next topic, the very important topic from ecology. Ecology, you are going to get mostly more than 10 questions from the ecology. In the recent NEET examination, you have more than 10 questions from the ecology. We have 3 chapters here: organisms and population, ecosystems, and the biodiversity. So what does ecology mean? Ecology is made up of 2 different terms called oikos, which is nothing but house, and logos, which is nothing but study, to study. So what does ecology mean? Ecology is the study of interaction of organisms with their environment is called ecology. Study of interaction of organisms with their environment, with the biotic and abiotic factors present in the environment is called as ecology. Okay, and the term ecology was given by Ernest Haeckel. And in the introduction part, it is given as Ramdev Mishra is the father of Indian ecology, and Eugene Odum is the father of modern ecology. Okay, so that's about the ecology. So in this particularly in this ecology chapter, we are going to discuss about the hierarchy of ecology. Mainly in the animal kingdom, we have discussed the hierarchy, like atoms combine to form molecules, molecules combine to form protoplasm, protoplasm to cells, cells to tissues, tissues to organs, organs to organ systems, that we have studied in the animal kingdom. Now the organ systems will combine to form organisms. Organisms of same species will combine to form populations. Populations will combine, different populations will combine to form communities. Different communities which interact with biotic and abiotic factors are called as ecosystems. Different ecosystems with a perfect boundary, they are called landscapes, and different landscapes combine to form biomes, and all the biomes together is called as a biosphere, nothing but the earth. So this is a hierarchy of ecology which we are going to study. Now in every biological organization, in every biological phenomenon, in every process, we can frame 2 types of questions. One is how question and as well as why question. How and why. How tells us about the mechanism and why tells us about the significance. Very important. How tells us about the mechanism and why tells us about the significance. For example, a bulbul sings the song in the morning. Now how does a bulbul sing a song? Because of the vibrating bone in its voice box, which is called syrinx. And why does a bulbul sing a song? To attract its mates for in the breeding season. So there is a mechanism, there is a significance, but for both the things. So how tells us about the mechanism, why tells us about the significance in this particular process. And similarly, we have some questions like why are night blooming flowers generally white? Because night blooming flowers, instead of wasting their energy to produce different colors, as they are not visible at all in the night time, so night time white is so much visible, so they will produce good fragrance to attract the insects for their pollination. So that's the reason night blooming flowers are generally white, and they produce very good fragrance to attract insects for pollination. And how do bees recognize the flower which has nectar? Because of the chemical impulses produced by this nectar. And why does cactus have so many thorns? You know, cactus have thorns because the cactus is a desert plant where the leaves are modified into spines, because leaves do have stomata. So when stomata are more, transpiration occurs and they lose more water. Already in desert there is a scarcity of water, so there is a chance of losing more water. So that's the reason these cactus plants the leaves are modified into thorns so that the water evaporation gradually decreases, transpiration decreases. So that is the reason cactus have thorns. And how does a chick recognize their own mother? This is a principle called imprinting. This is an animal behavior called as imprinting, by which they can recognize their mother. Okay, so this is how you can frame different types of questions for every biological phenomena or a biological processes that are occurring in our nature. Now, in the ecology, as I told you earlier, ecology is the interaction among the organisms and between its physical abiotic environment. In this, we are going to study 4 levels: organisms, population, communities and biomes. Organisms, populations, communities and biomes. Okay. Now, directly we are going with population. So, what is meant by a population? Group of similar species which can potentially interbreed, which are competing for the similar resources, is called as a population. Okay. So, these populations can be sometimes population not only is by the sexual reproduction, it is also by the asexual reproduction, right? Then this populations, generally if you go with some examples of the populations: cormorants in a wetland. Cormorants are the birds present in the wetland. Rats in an abandoned dwelling is also an example of population. Teakwood trees in a forest is an example of population. Bacteria in a culture plate, lotus plants in a pond, all these are different examples of population. Now, these populations and species, individual organism can cope up with the changed environment, but natural selection occurs in a population. Very, very important. Evolution. Okay. Then population ecology, a very good branch of science which deals with both population genetics and as well as evolution. So this is population ecology. Now, when it comes to population attributes, a population has so many attributes or characteristics to decide the changes, or whether the population is increasing or decreasing. We can decide with the help of the population attributes. The first population attribute is a birth rate and a death rate. So, birth rate and death rate. Birth and death can be seen in a species, an individual organism, but birth rates and death rates can be seen in a population. So, how can you assess the birth rate? For example, if in a pond there are 20 lotus plants last year and through reproduction 8 new plants have been added and the current population to be 28, how can you calculate the birth rate? Birth rate is equal to number of newly added by number of existing population. So, what is meant by existing population? The population which is already present in the lotus pond. So, that is number of newly added is nothing but 8 and number of existing population is 20, and 8 by 20 gives us about the birth rate, which is nothing but 0.4. Similarly, how can you calculate the death rate? The number of individuals dead at the period of time by the number of existing population. If you call, then definitely you can calculate the death rate. For example, Drosophila, out of fruit flies, out of 40, 4 were dead. So, what is the death rate? 4 by 40, which is equal to 0.1. This is a NEET question in 2024, a direct question which is given from the NCERT textbook, the same question. Okay. Then another attribute is sex ratio. What is meant by sex ratio? The ratio of males is to females or females is to males in a population is called a sex ratio. Okay, the male or a female. An example is given: 60% of the population are females and 40% are males. This is not always standard. Sometimes males may be more, sometimes females may be more. So that's called as a sex ratio. The next comes age pyramids. So age pyramids. So to assess the population, we are dividing the population into 3 different types of groups: pre-reproductive individuals, reproductive individuals, post-reproductive individuals. Pre-reproductive individuals are the people who mostly less than 18, they are not going to reproduce. Reproductive individuals are some 20 to 35 years of age group which are reproducing right now, and then post-reproductive individuals who already produce children, 1 or 2 children who can't reproduce right now. These are called as, they can reproduce but there is no need of reproduction. So these are 3 different types of individuals. In a population, in a ecosystem, if there are more pre-reproductive individuals and less post-reproductive individuals and less reproductive and post-reproductive individuals, now this will form a triangular pyramid, and this tells us about an expanding population because the pre-reproductive individuals are very, very high. Young generation is high. Now, if in a ecosystem the pre-reproductive and reproductive individuals are almost similar and then post-reproductive individuals are less, this will form a bell-shaped pyramid, which is nothing but a stable population. This is expanding pyramid is a triangular pyramid, whereas stable population gives us about the bell-shaped pyramid. Whereas the pre-reproductive individuals are less than the reproductive individuals, even post-reproductive individuals are also less than the reproductive individuals. This forms a spindle-shaped pyramid or an urn-shaped pyramid or a top-shaped pyramid, which tells us about the declining population. Spindle-shaped pyramid, urn-shaped pyramid or a top-shaped pyramid, which tells us about the declining population in that particular ecosystem. So by the age pyramids, we can decide whether the population is expanding or it is a stable or it is a declining population. Okay. Then, so to assess the population density, what is population density? What is the population, what is the number of population in that particular ecosystem is nothing but the population density, which is designated by the letter N. To assess the population density, there are some factors to assess it, whether it may be adverse weather conditions or the predation pressure, okay, or availability of food and space. So to assess the population density, we have different methods to assess the population density. First one is direct count. Sometimes by directly counting, we can totally count the total number of organisms in that ecosystem. For example, Siberian cranes. Siberian cranes coming to the Bharatpur Islands, the Colorado National Parks, it can be easily counted because the count will be less than 10. It's a direct count. But sometimes we cannot count the total number of Chlamydomonas which are present in a pond. Chlamydomonas, because these are algae, unicellular organisms which are present on the surface of the pond. Can we count them? No, we cannot count them. In such conditions, we will go with the biomass of that particular organism, the dry weight of all the organisms. Similarly, counting sometimes becomes meaningless. Counting sometimes it becomes meaningless when we are discussing about banyan tree and a Parthenium plant. A big banyan tree, a huge banyan tree with large canopy, and there are small Parthenium plants below this banyan tree. The banyan tree is very huge, it has taken some thousands of years to grow, whereas Parthenium plant is a small weed. Parthenium hysterophorus carrot grass is a small weed which takes within few days to develop. Even if there are 200 Parthenium plants, I cannot de-estimate the value of the banyan tree by simply saying it has only one, because banyan tree has huge, it's a huge ecosystem, it's a tree ecosystem. It has so many insects, so many caterpillars, so many birds, so many animals living on the banyan tree. It has a huge experience because it is formed around some thousands years. By saying simply it has one, I cannot de-estimate its value by saying there are 200 Parthenium plants. No. So in that particular situation, counting becomes meaningless. So there we can assess this with the help of biomass, or we can also go with percent cover. Percentage cover of this banyan tree sometimes. Sometimes the bacteria in a culture plate also can be assessed with the help of biomass, because we cannot count the bacteria. They are invisible to our naked eye, so we can assess with the help of the biomass. Sometimes relative density is being used here, relative density, because relative density, the fish caught per trap. In a pond, right, by getting the fishes in a net, the number of fishes in a trap can be used to estimate the total number of fishes which are present in the pond approximately, and that principle is called as relative density. Sometimes indirect count indirect count. So, without looking at an organism, we can count the organism like tiger census in the national parks, tiger census. So, how can you count the tiger census? By pug marks or footprints of the tigers and as well as fecal pellets of the tigers, because pug marks, even though the tigers look similar, the prints of those tigers are different. The pug marks are different because even though we are similar, we all have same fingers and same hands, but our fingerprints are quite different. Everyone will have different fingerprint and no two organism, no two people will have the same fingerprints. Similarly, tigers, the pug marks, they are quite different. And the fecal pellets also, they are different because it depends upon the food which is eaten by the tigers. So, by assessing the fecal pellets and the pug marks, the footprints, we can count the total number of tigers, because we cannot go to every tiger and say, hi, you are number 1 and number 2. It's not possible. So, the counting of tigers can be done with the help of fecal pellets and the pug marks. So, this is how we can count, we can have the estimation of the population density by different types of methods. Now, the population growth. So, the population growth depends on various factors like food availability, then predation pressures and adverse weather. Sometimes availability of food decides the population, adverse weathers decide the population, even predation, because if the organisms are killing them, it also decides the population. But there are 4 major factors which mainly help in increasing or decreasing the population, whether it is flourishing or declining. What are the 4 factors? Natality, mortality, immigration, emigration. Natality, natal is birth. So, number of births added to the initial density is called natality. Number of deaths in that particular population is called mortality, mortal is death. Immigration, the people who are coming from some other area to the present population of same species is called immigration. People who migrate from one particular present area to some other place is called as emigration. So, if you look at this particular picture, the natality is represented by capital B, and natality, the arrow mark is indicating towards the population density, because natality is increasing the population, represented by a positive sign. Mortality, the arrow mark is towards away from the population density, because mortality will decrease the population density, represented by a negative sign. Immigration is increasing the population density, the arrow mark is towards it and it is represented by a positive sign. And emigration is away from the population density and is represented by a negative sign. So, natality and immigration are increasing the population density, mortality and immigration are decreasing the population density. Now, if you plot a graph, or if n is the population density at time t, then density at time t plus 1, tomorrow or day after tomorrow or next year, whatever it may be, can be assessed by a formula. N of t is equal, t plus 1 is equal to n of t plus b plus I minus d plus e. If b plus I is greater than d plus e, then the population density gradually increases. If b plus I is less than d plus e, the population density gradually decreases. If b plus I is equal to d plus e, the population density is stable or remains stable, right. So, in a normal condition, these statements are very important here. Under normal conditions, births and deaths will decide the population growth. Whereas, if a new habitat is been colonized, a new habitat is been formed, immigration, very important, immigration plays a very important role in population growth. So, that is how we can assess the population factors. Now, based on these population factors, we have 2 different models, growth models. One is called as exponential growth, and the other one is called as logistic growth, Exponential growth is also called as geometric growth. Exponential growth is also called as geometric growth. So, what is exponential growth and geometric growth? Exponential growth is mainly because of unlimited resources. Exponential growth is because of unlimited resources. So, when the resources are unlimited, according to Darwin, when the resources are unlimited, then the organisms will continuously reproduce and increase their population. So, that is nothing but exponential growth. Whereas logistic growth is because of limited resources. When the resources are limited, organisms cannot utilize their fully innate potential, fully biotic potential or reproductive potential, because resources are not available. So, the growth is limited. If you plot this exponential growth in a graph where for population density with time t, the growth is represented by a J-shaped curve because the population keeps on increasing. Whereas if the same graph is plotted for the logistic growth for n and the density at time t, the population growth first it slowly increases, which is called as lag phase, then it increases rapidly, which is called as log phase. This is lag, this is log or acceleration, because resources are more, the population increases rapidly. Now when the resources slowly decrease, it causes decrease in the population, so it is called as deceleration phase. Now when the resources completely get ended up, it results in stable phase, which is called as asymptote phase, which is a stable phase. Why this asymptote phase? Because the resources get ended up. The place where the resources, the stage where the resources get ended up, it is represented by the letter K, which is called as nature's carrying capacity. Nature's carrying capacity will decrease the will stop the growth because the resources will become limited. Now, based on this, so it is giving a sigmoid shaped curve. Logistic growth is represented by a sigmoid shaped curve or S shaped curve, whereas exponential growth is represented by a J shaped curve. Now when we derive a formula for exponential growth for difference in population density for in time t, this is given as B minus D birth rate per capita death rate minus per capita birth rate minus per capita death rate into population density. Now this per capita birth rate minus death rate can be taken as R, which is called intrinsic rate of natural increase. What is this R value? R, intrinsic rate of natural increase, is a factor which decides the growth of the population, because if resources are unlimited and in ideal conditions, ideal environmental conditions, all the environmental conditions are ideal which are favorable for the increase of the population growth, then the R value is high. If the R value is high, the population density is also high. So what does this R mean? Intrinsic rate of natural increase tells us about the availability of resources and ideal environmental conditions and impact of the biotic and abiotic factors on the population growth. Because if biotic and abiotic factors are not favorable, then population growth will not happen. If all these are favorable, then the growth increases. It means that there is a very high R value. Or this can, so dn by dt is equal to Rn. If you go with an integral formula, this can be taken as n naught is nt is equal to n naught e to the power of RT. Nt is population density at time t, n naught is population density at time 0, E is base of natural logarithm, R is intrinsic rate of natural increase, and T is time t. This was a question in 2024. What does E represent in this particular equation? E is base of natural logarithm. This is about the equation for exponential growth. Whereas for equation of logistic growth can be given like this: dn by dt is equal to Rn into k minus n by k, that is nature's carrying capacity. Or I can take like this: dn by dt is equal to Rn into 1 minus n by k. This is the growth equation for the logistic growth. Okay. So out of these two, exponential growth and logistic growth is said to be the more realistic growth, because no individual will have unlimited resources at his disposal. No one. No individual, no organism will have unlimited resources, because as Darwin says, even a slow growing animal like elephant will grow continuously on absence of checks. If the checks is absent, if the control is absent, even elephant also grows faster. But no individual will have unlimited resources. So that's why, which is said to be the more realistic one, logistic growth is said to be the more realistic one. And the R value, you have to remember the R values. R value for Norway rat is 0.015, the desert rat which never drinks water in its lifetime, and floor beetle, because the beetle which has much resources, its R value is 0.12. And human population in India, the R value is 0.0205, and right now it is around 0.84. Okay. So this is how the population growths can be seen, population exponential growth and as well as the logistic growth. And there is a story explaining about the exponential growth, about the king and minister story, about the chess game. The minister, the king with this ego, the minister gives a bet that if he loses the game, he have to give the wheat grains in every chessboard, in every square of the chessboard, increasing them, squaring them in the next square like this 64 squares. The king thinks it is very easy, but once the king loses the game, if he is keeping on wheat grains in these chessboards, once the half of the chessboard has been completed, the wheats totally in his kingdom would have been completed. Okay, because this is exponential pattern or this is a geometric pattern. Okay, right. Then life history variations. Life history, there are some changes in the organisms in their life histories depending upon the availability of resources. Some organisms, they breed only once in their lifetime. For example, Pacific salmon fish and bamboo, depending upon the resources. While some organisms breed many times in their lifetime, mostly birds and as well as mammals. Some organisms will produce large number of small sized offsprings like oysters and pelagic fishes, because even though they produce large number, because of overproduction, Darwin's theory we have seen, overproduction, constancy in population, struggle for existence. So even though they produce large number, they are very small, majority of them will die, only few will survive. Whereas some animals will produce a small number of large sized offspring but less in number, their lifespan will be more. These are about some life history variations. So these life history variations, mainly why do these life history variations? Because to maximizing their fitness, right? To live, to evolve, to survive, they have different types of life history variations. Okay, then next comes to the population interactions. So no organism can survive individually. Every organism has to depend on some other organism for their survival. So in that, we talk about different types of interactions. We have two different interactions like intraspecific and interspecific interactions. Interspecific interactions are between two different species. Intraspecific interactions are between the same species. Now here in this chapter, we are going to discuss mainly about the interspecific interactions between two different species. Now mainly we discuss here about the different types of where in mutualism, both the organisms get benefited. Plus and plus sign is given, plus is for benefit, minus is for harmful, 0 is for neutral. Whereas in predation, in competition, both the organisms are harmed, so it's both are minus. Predation, predator gets benefited, prey gets harmed. Parasitism, parasites get benefited, host is harmed. This is parasite. Parasite is benefited, host is harmed. In predation, predator gets benefited, prey gets harmed. Okay, whereas in commensalism, one gets benefited, either is neutral, neither benefited nor harmed. In commensalism, one gets harmed, the other is neither benefited nor harmed. It's an example where the antibiotics have been produced between Staphylococcus bacteria and Penicillium fungi. Penicillium fungi, Penicillium notatum in a Petri plate. When the Alexander Fleming was working here, the Staphylococcus bacteria, these Staphylococcus bacteria, when a penicillium spore fell on this bacteria, the fungi developed here. When the fungi developed, there were no bacteria surrounding this fungi, because this fungi started producing a chemical called penicillin, and this penicillin was acting as an antibiotic which is killing the surrounding bacteria. So by this, the discovery of the antibiotics has been done, which are produced by one microbe which kills the other microbe. So here in this association, penicillin is neither getting benefited nor harmed, but Staphylococcus has been harmed. So that's why one is harmed, it is minus, and other is neither benefited nor harmed, it is 0. This association led to the discovery of antibiotics. One of the previous NEET question, which of the following association led to the discovery of antibiotics? Which is amensalism. Okay. Then, now in this, so we have the different types of associations, and please see this statement: predation, parasitism and commensalism, the interacting species live closely together. Please follow the statement from your NCERT. Predation, parasitism and commensalism, the interacting species live closely with each other. Okay. Then, now when it comes to predation, predation is an association with where one is benefited and the other one is harmed. Predator is benefited, other one is harmed. So always when you talk about predation, we don't talk about lion or the tiger, which is always a carnivore. Sometimes even a herbivore, the sparrow is also a predator. What is a sparrow? Because sparrow is feeding on the sparrow eating the seeds is also a predator. because it is eating on the plant. Okay. In all the animals eating plants are categorized as herbivores. In broad context, they are very different from the predators. If these predators act as conduit. What is meant by conduit? It acts as a pipeline for the transfer of energy in a food chain. Say for example, grass, grasshopper, frog, snake and hawk. So now grass is the plant which is utilizing solar energy and preparing its food with the help of photosynthesis. If we stand in the sunlight, we can't get any energy. But we get energy when we eat the plants, because plants are helping in conversion of this radiant energy into the chemical energy, available energy for us. But how do we get this energy? When we eat the plant. In the sense, we are a predator who are eating the plants. So predation helps in the transport of energy from one level of food chain to the another level. So it is called as conduit. Then predation helps in biological control. So what is meant by biological control? Here in this particular paragraph, you are going to look at the biological control. So what is meant by biological control? One organism keeping other organism under control is called biological organism. We have seen the Gambusia fish which is controlling the mosquito larvae. Similarly here in the biological control, we are going to discuss about the cactus plant in Australia in 1920s. There were the cactus plant, prickly pear. This cactus plant was growing enormously. Now how to reduce this cactus plant? To reduce this cactus plant, because no herbivore is going to eat the cactus because it has a lot of thorns. Now, but the scientists found that there was a predator called Cactoblastus cactorum. There was a caterpillar larva called Cactoblastus cactorum. This is a larva this is a caterpillar which is feeding on the cactus plant. So the scientists developed these larvae and then they just allowed these larvae to eat away the cactus plant. So this is also an example for biological control. Now the pesticides which we are using in the farms, the biopesticides, it is also an example of biological control, biopesticides. Now the next is, if the predator overexploits its prey, okay, before that the predation acts as the prudent, or it is helping in controlling its prey, because if the predators are not available, then the prey population also gradually decreases. Here for example, if you get in the American Pacific coast of rocky intertidal communities of American Pacific coast, the starfish pisaster is an important predator. But if the starfish pisaster is removed from that particular area, the number of the 10 species of invertebrates became extinct. 10 species of invertebrates became extinct. So predation is also helping in maintaining the population of the prey. Okay. Then, so predator is a prudent, because if the predator overexploits its prey, then the prey population gradually decreases, automatically the predator population also decreases. So predator will never overexploit its prey. So that's why it is called prudent. We call the term which is given here, which is called as prudent. Sometimes the species, the prey population will have some adaptations to survive with the predators. For example, camouflage. Some insects and frogs undergo camouflage. They change their colors to avoid from the predators. Sometimes the monarch butterfly, example monarch butterfly, it becomes distasteful, because during its caterpillar stage it feeds on a poisonous weed. When it feeds on a poisonous weed, it becomes distasteful. So when it is distasteful, no other bird or any other organism is going to eat it. Now sometimes plants, how do they get protection from the predators? Because even plants also have the protection, because nearly 25% of the insects are phytophagous. What are phytophagous? The insects which are feeding on the plant sap. Now how do the plants get the protection from the insects and as well as animals? The plants, some of the plants will produce thorns and spines called acacia and cactus. Some of the plants will produce many chemicals. When the organisms eat these plants, their digestive systems and reproductive systems are getting affected, like calotropis. Some of the plants, because calotropis store poisonous cardiac glycosides which are disturbing the digestive system and reproductive system of the plants. And some of the plants will produce secondary metabolites like nicotine, caffeine, quinine, strychnine, opium. These secondary metabolites also protect the plants, because when the herbivores eat the plants, these secondary metabolites which are going to affect their body. So that's why the herbivores are not going to eat these plants. So this is how the plants are getting protection from the grazers and the browsers. Okay. Then the next association is competition, very important association. Competition is an association where both the species are being harmed. According to Darwin, interspecific competition is a potent force for organic evolution. Interspecific competition, competition between two different species is a potent force for organic evolution, right? Now, always competition does not occur between the same species. Competition sometimes occurs between the different species also. For example, shallow South American lakes visiting flamingos and resident fishes compete for zooplankton. Sometimes even resources may not be limiting, sometimes resources are very high. But still, if a competitor, if one of the competitor is having high competitive ability than the other one, interference, he is interfering with the other one, the other one could not eat properly. So that is also called interference competition. So always competition does not occur when the resources are limited. Competition even occurs when the resources are unlimited because of interference competition. Okay. Because in this interference competition, the organism whose fitness is less is going to be eliminated. Based on this, Gauss have done experiments with the help of the paramecium and as well as some other organisms. He have given a principle called competitive exclusion principle. It is not always conclusive, but there is a strong evidence for this competitive exclusion principle where because of competition, one of the species totally got eliminated. For example, in Galapagos Islands, there is a tortoise called Abingdon tortoise which was totally eliminated within a decade, within 10 years because of the goats, as these goats were introduced into the island. Goats will have very high browsing efficiency, very high browsing capacity. So because of this high browsing capacity, tortoises will have low browsing capacity. So totally tortoises were eliminated from this islands, Abingdon tortoise. So this is called competitive exclusion. Sometimes competitive release. What is meant by competitive release? If a superior competing species is released from that particular area, removed from that particular area, the inferior competing species will automatically develop. It grows. That is called competitive release, which is given by Cornell's elegant field experiments where in the rocky intertidal areas of Scotland, the superior barnacle Balanus and the inferior barnacle Cathamalus, if Balanus is removed, the Cathamalus is going to survive. This is called competitive release. Sometimes there is a coexistence between, because Gauss competitive principle says that one of the organism is going to get eliminated if both are competing for the resources. But sometimes there is a coextinction between sharing of resources between the organisms, which is called as resource partitioning. Resource partitioning. What is resource partitioning? If the resources are being shared between the organisms, then easily they can survive. The best example for that is warblers. Warblers will share their resources on a same tree, which was given by MacArthur. This is called coexistence. Both will live together by sharing their resources given by MacArthur. But these warblers will differ in their foraging activities. Foraging in the sense food collection. They differ in the food collection. Some will eat in the lower parts of the tree, some will eat in the inner parts of the tree, some will eat at the top layers of the tree, some will feed on insects in the summer season and fruits in the winter season, some will feed on fruits in the summer season, insects in the winter season, like this. So they will change their behavioral as well as foraging patterns so that they can survive together. This is called coexistence, and as well as it is also called as resource partitioning. Okay, this is about the competition. Then parasitism, you all know parasitism, parasite, and this is an interaction between a parasite and as well as a host. Parasite gets benefited, host definitely gets harmed because host is providing free lodging and boarding for the parasite. The parasites, in order to survive in the host, always the parasites are host specific. But the host always keeps on rejecting the parasite. But in order to survive in the host, the parasites will have some adaptations. What are the adaptations? Loss of unnecessary sense organs, presence of adhesive organs or suckers to attach to the host, loss of digestive system. The parasite loses its digestive system, it absorbs food from the surface, and high reproductive capacity, because if it produces more and more organisms, it can easily survive inside the host body. These are the adaptations of a parasite living inside the host. So there are some different types of parasites. See, for example, some parasites will have more than 2 hosts in order to survive. For example, human liver fluke, the trematode parasite. Remember, trematode parasite depends on 2 intermediate hosts, a snail and a fish, to complete its life cycle. So, complex life cycles also is one of the type of adaptation. Malarial parasite needs a vector mosquito to spread the other host, but they do not harm the host, right sir. Then these parasites, what they are doing, these parasites harm the host, they reduce the survival of the host, they reduce the growth and reproduction of the host and reduce the population density of the host, and as well as they prone them to predation, making it physically weak. Okay, and these parasites will never, when the host evolves, the parasite also evolves. There are no friendly parasites, all the parasites harm us. Parasite, there are some ectoparasites, types of parasites if we talk about. Ectoparasites, parasites which live on the surface of the body, right. For example, the lice on the humans, ticks on the dogs, and copepods on the fishes. These are all ectoparasites, and there is an ectoparasite, plant parasite called cuscuta, which is found on the hedge plants. This cuscuta will lose its chlorophyll, it loses its leaves, but it has hostoria which can penetrate into the host plant and get the food and the water. Right. The female mosquito is not considered as a parasite because it doesn't need blood for reproduction, it just stays for a shorter time on our body, and then so it is not considered to be a parasite. Right. Then endoparasites which lives inside the body of different organs in our body, and these endoparasites, their morphological and anatomical features are greatly simplified while emphasizing their reproductive potential. The reproductive potentials will be very high, but morphological and analytical features will be gradually reduced. This is very, very important. Then brood parasitism. Brood parasitism is between a crow and the cuckoo. Cuckoo, almost it lays eggs which are almost similar to the crow's eggs, and the cuckoo, when it lays eggs inside the crow's nest, the eggs are almost modified, eggs are almost evolved that this egg exactly resembles the crow's eggs with markings and the size. Now once the cuckoo bird hatches out, when the cuckoo bird hatches out from this cuckoo bird is going to push the eggs of this crow outside. The eggs are being pushed away by this cuckoo bird. Now these eggs of the crow will drop down and the eggs will got broken, and the crow's population is going to get reduced. So here cuckoo is acting as a parasite and crow is acting as a host, which is called as a brood parasite, and the breeding season of this cuckoo is from during the course of evolution. Parasitic birds like cuckoo, they develop the color and size exactly same, and during the breeding season this complete process takes place. This is about the parasitism. Then next comes to mutualism. What is meant by mutualism? Mutualism is an association where both the organisms will get benefited. Before mutualism, we have okay, let me tell you about mutualism. Mutualism is an association where both the organisms will get benefited. Now in the mutualism, for example, let us go with the lichens, algae and fungi. Lichens, algae will produce the food material and fungi will produce the space. It's an association. And mycorrhiza with higher plants, mycorrhiza with the roots of higher plants, it is also an association between both mycorrhiza and the roots where both are getting benefited. Then the most important association here, we talk about the wasp and the fig fruit. The fig fruit will provide a suitable site for the wasp to lay its eggs, and also fig fruits provides food for the developing larvae. What the wasp, right, the wasp pollinates the fig flower, inflorescence, fig inflorescence. When it is moving into the fig inflorescence and coming out, it pollinates the flower. So the fig is pollinated by the wasp, and the wasp develops its larvae. Wasp lays its eggs and develops its larvae inside the fig fruit. The wasp makes small holes, the female wasp makes small holes inside the fig fruit. When it is going into the fig fruit, it loses its wings. Now when it enters into the fig fruit, it lays so many eggs inside the fig fruit, and then these eggs will develop into larvae, and the seeds which are present here will be food for the developing larvae. Now these larvae, first the males will develop here, males will copulate with the females and release the sperms into the female body, and then the males will come out of the fig fruit by making some holes inside the fig fruit, and the males will die. And these holes, after the females get matured, the females come out of these holes and will such a new fig fruit, and again enter into the fig fruit and pollinate the fig inflorescence. So this is how both are living together. Fig cannot live without wasp, wasp cannot survive without fig. Right. So when fig becomes extinct, the wasp also becomes extinct, and if wasp extincts, fig also extinct. This is called co-extinction. It's a mutualistic association. Similarly, we have some interaction between the bumblebee and the orchids. The orchids and bumblebee, orchids will show a very wide diversity, and the bumblebee, the orchids develop in such a way that their flower exactly resembles the female bumblebee. So for example, if we take a look at it, the orchid offeris employs sexual deceit. What is meant by sexual deceit? Sexually, it is deceiving the male bee because one of the petal of its flower bears an uncanny resemblance to the female bee. It exactly looks like the female bee in size, color, and as well as markings. So the male thinks it as a female, and then the male copulates with the flower, which is called a pseudo-copulation, where in this pseudo-copulation process, the male is transferring the pollen grains. So pollination occurs to this flower. So the flower is employing the sexual deceit, the male is doing the pseudo-copulation, both are benefited, and then this is also an example for mutualism. If at all the female bee color changes, the nature in the female bee, if there is any change in the markings, if there is any size in the marking, size and color of the female bee, the flower automatically evolves in order to change exactly like the female bee, so that because if the flower doesn't change, the male will never get attracted to the flower. So when the female bee changes, the flower automatically evolves, and this principle is called as co-evolution. This is called co-evolution. So this is how you can see some examples of mutualism. Now when it comes to commensalism, in the commensalism in association where one gets benefited and another gets harmed. In the commensalism, we see the epiphyte on the mango tree. An epiphyte is a plant which develops on the mango tree. It doesn't take any nutrients or from the mango tree, but it just takes the shelter from the mango tree. So epiphyte is getting benefited, but the mango tree is neither getting benefited nor harmed. Similarly, cattle egrets. What are cattle egrets? The birds which are present near the grazing cattle. So when the cattle are moving, because of the hooves of the cattle, the grass, when the grass is moving, the insects which are present in the grass will be coming out, and these insects are eaten away by these egret birds which are moving with the help of this or moving on the cattle. So cattle egrets are getting benefited, cattle neither gets benefited nor harmed. Similarly, we have the fishes, sea anemones and clownfish. Sea anemones and clownfish. Sea anemone, you know, sea anemones with nidoblasts. The clownfish stays inside the sea anemone. This clownfish has a jelly coating which can be protected with the help of the sea anemones, but there is no other organism which comes and attacks this clownfish. So the clownfish is getting benefited with the help of the sea anemone. Sea anemone neither gets benefited nor harmed. There is one more association called barnacles on the whales. Whales will have barnacles on their body. These barnacles, arthropods, will stick on to the whale and they move with the help of this whale. Wherever the whale moves, the barnacles also move with the help of this whale. So the barnacles are getting benefited, whale is neither getting benefited nor harmed. So this is called a commensalism association. And we have seen amensalism where the Staphylococcus bacteria and the fungi association where Staphylococcus is being harmed, fungi is being neither harmed nor benefited. This is called as an amensalism minus N0. So these are all the different types of interactions, interspecific interactions, where we are discussing about so many examples and very important. Definitely have a question from the interspecific interactions from these organisms and populations. So this is a summary where in the summary you are going to see all the population growths, and you can see all the associations which are present here. Competition, both get suffer. Predation, parasitism, one benefits and other suffers. Commensalism, one benefits and the other unaffected. Amensalism, one is harmed, other unaffected. Mutualism, both are benefited. Okay, so this is about the organisms and the population chapter, from which you are going to get definitely 2 or 3 questions in the final NEET examinations. From biotechnology and applications in medicine, you are going to get 1 question. From organisms and populations, you are going to get 3 questions. 3 to 4 questions from this particular chapter. Very important chapter. Read every line of NCERT so perfectly. Nothing much differences, nothing much deviations from this chapter from the last NEET examinations. So if you are perfect with the NCERT in this particular chapter, you can easily answer those questions from the NCERT. So keep reading NCERT many times, go thorough with the NCERT, and all the best for your NEET examination, the coming NEET examination. Do well, you can do your best, you are the best and you can do your best. All the best, God bless you.